Students, in this module we'll be studying about the test cross. Fine. So students, first of all we should know that what exactly the test cross is and why it is being used up. Now the cross is made between a hybrid, fine, that is the two different alleles, and one homozygous recessive plant or the parent. Now it is known as test cross, and why it is used up? To identify the genotype of the hybrid. So suppose we want to know the genotype. So how will we be able to know this thing? With the help of the test cross. Fine. So you can see here, this one is the hybrid tall, students. Fine. And this one is the recessive dwarf. So now talking about their genotype, that is the capital T small t, and this one is the small t small t. Now what about their gametes? Here you can see I am having a capital T and small t, and here I am having a small t. So students, when this in F1 generation I am talking about, when this capital T get fused up with this small t, so here you can see the capital T and small t is formed, where capital T is able to express itself, and that is why it is known as tall one. So the plant is tall here, the parent is tall. And talking about when the small t of this hybrid tall plant, when get fused up with the gamete of the recessive dwarf parent, then here you can see small t is formed, that is the dwarf parent. Fine. So here talking about their ratio, students, that is the phenotypic one. Fine. And the genotypic is also 1:1, and phenotypic is also 1:1. Now students, talking about the incomplete dominance. This incomplete dominance is basically seen in, for you can say the example is snapdragon. We have dog flower plant. Fine, you can see here the picture. And another example is Mirabilis jalapa, that is 4 o'clock plant. So now students, Correns discovered this incomplete dominance, that is in Mirabilis jalapa, and it is also called as partial dominance, or we can say the semi-dominance. Fine. So when experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the parent and was in between the two. So students, the inheritance of flower color in the dog flower, like we have snapdragon, is a good example to understand the incomplete dominance. Fine. So the inheritance in which allele for a specific character is not completely dominant over other allele, and that is said to be incomplete dominance. Fine. And the result that we are getting here is between the two. Fine. So it is a mixed up kind of thing. So you can see here, students, I would like to show it to you with the help of this. See, now talking about their genotype. So you can see here we have the capital R, fine. This is a different flower that we are having. Fine, that is the, you can see the parent is, I'm showing parent with the genotype with the red color line. So you can see her parent, when it is red in color, the genotype is represented by the capital R allele, student, and the parent is white in color. So the parent is white in color and it is represented by the WW, capital W. Fine. Now when this red, that is the parent generation. Fine. So when it is they are fused up together, their gamete is capital. This capital R is the gamete of the red parent, and this W, that is the capital W, is the gamete of the white parent. So what is formed here? You can see here, students, the thing that is formed is the pink color. It is neither red nor white, but it is something in between. Fine, this is what the pink color is. I hope I am clear, students. Now, this is the F1 generation, which is the first filial generation, right? Talking about their F2 generation, that is the filial 2 generation, that we will be getting only if we will do the selfing. Fine. So, you can see here, students, if we'll do the selfing, so you can see the F2 generation, you can see the gametes that are formed is capital R and capital W. Fine. Similarly, there will be the female gametes also, the capital R and capital W. When they are both like pollinated together, so you can see when this capital R get fused up with this capital R, so you can see this red is formed. Similarly, when this capital R gets fused up with this W, then it is, see, I'm going to show you with the help of the color. Look, these are the gametes, fine. One represents the male, another represents the female. So you can see here, these are the gametes RW and RW. So see students, now very very important, when this R get fused up with this R, so you can see this red is formed. Fine, red is the color that you can see is of the parent only, one of the parent color. And similarly, when this R get fused up with this W, which is white color, so you can see neither red nor white has been formed, but something is formed that is in between, and that is the pink color. Similarly, students, the second gamete, when it get fused up with this capital R, this pink is formed. Fine. And when this fused up with this capital W, that is the white is formed. Okay. So now you can see here, students, that the phenotypic ratio is 1:2:1. And talking about their genotypic ratio, that is also 1:2:1. Okay. Now we should know that what is codominance. Very important concept, students, very very important. Now both the alleles for a character. Now one character is having two alleles, students. They are dominant. So when they are dominant and they are expressing its full character, that is said to be the codominance. Fine. So a good example of different types of red blood cells that determine the ABO blood grouping in human being. Now students, this ABO blood grouping, they are controlled by the gene I, and the plasma membrane of the red blood cells has sugar polymer that protrudes from its surface, and the kind of sugar is controlled by the gene. Now the gene I, this one is the gene I, it has three different alleles, students. Fine. It has three different alleles, and those are IA, IB and I. So these are the three different alleles, students, which are very very important. Now the alleles IA and IB, they produce a slightly different form of the sugar, while allele I does not produce any sugar. So this I, it does not produce any sugar. Fine, does not produce any sugar in it, or we can say do not produce sugar. Fine, it did not produce sugar in it. Now students, the ABO locus is located on chromosome 9. Fine. And I does not produce any antigen. But talking about IA, that is responsible for the production of antigen A, and IB responsible for the production of antigen B. Now a very important thing that happens here is, students, I would like to show you, is that these are the blood groups, and talking about their genotype, so you can say the A blood group is having the genotype IA, or it can be IA or simple I, and it does not produce any sugar. So I gene does not produce any sugar, students. Talking about B blood group, then IB, or we can say IBI. And AB blood grouping, you can see both are there. IA is also there and IB is also there. But O blood group is when I and I genes are there. Now students, see, two different alleles are there, I and I. Now see, because humans are diploid organisms, fine, and each person possesses any two of the three I gene alleles. So IA and IB are completely dominant over I. In other words, when IA and I are present, fine, so only IA expresses. So you can see with the help of this chart, students, because I does not produce any sugar. So when IA and I are present, only IA expresses. Similarly, students, when IB and I are present, IB expresses. Fine, because it is dominating one here. From this I and plus I does not produce any sugar. But when IA and IB, they both are present together, then what will happen, students? They both express their own type of sugar. And this is what the codominance is. So when both the alleles are present, that is with IA also and IB also. So at that time, the AB blood type of offspring is formed. Fine. I hope I am clear, students. So this was about the codominance. Now moving on to the next one, that is inheritance of two genes. See students, we have already discussed the inheritance of one gene, that is with the help of the monohybrid cross. Now talking about the inheritance of two genes, so this is also known as the Mendel's second law, that is the law of independent assortment, which states that factors for different pairs of contrasting character in a hybrid distributed independently during gamete formation. So when gamete formation is there, so different pairs of contrasting characters which are present in a hybrid, they basically distribute uh, or we can say assorted independently during the formation of gametes. Now students, the cross between two parents which differs in two pair of contrasting character, that is said to be the dihybrid cross. Fine. So you can see here, this one is the dihybrid cross students. Look here. So talking about the parents, fine. So you can see we have taken two contrasting character. One is round yellow, another one we have is the wrinkled green. Fine. So you can see here, see I would like to show you with the help of different colors. So for yellow I am taking yellow. This one is the round yellow and this one is the wrinkled green. Fine. So you can see here the genotypic for round yellow is capital R, capital R, capital R and capital Y, capital Y. Talking about the wrinkled green, it is small r and small y, small y. Fine. So and now talking about their gametes. This is basically students, this is for the simplification we have taken it. Like for the round we are taking RR, but for the wrinkled we are not taking WW, otherwise it will be like you will be confused. Fine. So that is why these initials have been taken. So talking about their gametes now students, so you can see here in this round yellow you can see we are having the capital R, capital Y, and here in this wrinkled green we have the small r, small y, and this is what the round yellow. So we can say in F1 generation the whole of the generation was round yellow. Fine. The complete generation was round yellow. Now students, talking about the complete dihybrid cross. So you can see here this one is the second generation. So you can see the gametes that has been formed. Fine. So here I would like to show it to you. See, we are having these capital R, fine, and what else we are having here? See, we are having the capital R, small r, and capital Y, small y. Fine. So I'm going to take these gametes only, and capital Y. See, capital R, capital Y, small y. Fine. So see, here four different gametes have been formed. Suppose this capital R get fused up with this capital Y. So you can see capital R, capital Y has been formed here. Look this thing very carefully. Similarly, when this capital R get fused up with this small y, so we get this capital R and small y. Fine. Similarly, students, when this small r get fused up with this capital Y, fine. So we got this small r and capital Y. And the last one is when this small r get fused up with this small y, we have got the small r and small y. Fine. So these are the four different gametes that has been formed. These are the female gametes. Similarly, here talking about the male gametes, the same gamete, because selfing has been done, students. Fine. This thing we have already studied in class 10th. Now see, when these are the different gametes. These are the male gametes. So male gametes I am circling with the green one and the female is with the red one. So you can see when these together are fused, basically these two. Fine. So this thing is formed. That is the capital R, capital R, capital Y, that is the round yellow. Fine. And similarly, when these two are fused up, so you can see here that capital R and capital Y, small y is formed. But here it is known as round yellow only, students, because the Y yellow is the dominating one. Fine. So similarly, you can do the rest of this these Punnett squares column, students. Similarly, when this capital R, capital Y get fused up with the small r, capital Y, this thing is formed, and then this thing, and then so on. Fine. So you can see here, students, in the end, we are left with the phenotypic ratio, that is the 9:3:3:1. Fine. So the resulting, you can see the genotypes and phenotypes. We have 9 by 16 is round yellow. Fine. Then we have the round green, and then we have the wrinkled yellow, and then we have the wrinkled green. 3 by 16 and 1 by 16. Now talking about the dihybrid test cross, students, we have already done this thing. So when F1 hybrid is crossed with recessive green wrinkled pea plant, fine. So this is what about the dihybrid crosses, where we take the two pair of contrasting trait, where the recessive green wrinkled is small r, small y, and the gametes are formed, that is the small r, small y. And talking about the F1 hybrid, that is the round yellow, this one is the F1 hybrid, students, fine. These are the gametes, fine. So this is the dihybrid test cross. So these are the gametes that has been formed, students, and this one is the recessive green wrinkle, the small r and small y. So you can see here, when we fused up with this recessive green wrinkled this gamete with these gametes, so this thing is formed, and here we can actually use this dihybrid test cross. So the phenotypic ratio becomes 1:1:1:1. So this was all about today's module, students.